Hi, well, welcome to TCT 2020 virtually, and I'm joined by Jeff Moses. We're talking about embolic protection during TAVR. Talk to us, Jeff, about the REFLECT-2 study.
Well, this was a study looking at a new device that was called the TriGuard 3, which is a little different from what people are used to with the Sentinel. This is a device which actually is inserted through the femoral access site through an 8 French sheath where you put the pigtail. So it's very similar to the usual treatment algorithm. And it just sits as a mesh that covers all 3 vessels, again, unlike the Sentinel, and presumably protects the entire brain from embolic debris. It doesn't capture it, it's a deflector.
How big of holes are in that mesh? I mean,
it's about 100 and change versus 145. That's the pore size. Got to get blood to the brain somehow.
Yeah, I can't stop. I can't stop that.
Yeah.
So, what was the design of the study, Jeff?
Well, these studies, as you know, are very complex, and frankly, it's been a journey on this, and this was, of course, designed before the Claret was approved. And it's 2-phase study. Number 1 is looking at safety against objective performance criteria, though we did have an active control, which wasn't powered to, but just for comparison. And the other part was efficacy, which was compared to a control group, which is a little exotic, only that we had our active controls, but we also had a first phase of the study that we kept blinded, and we employed those controls as well in order to maximize our power. That was, again, a hierarchical endpoint combining clinical and neurologic and MRI criteria. It was done by the Finkelstein-Schoenfeld method, which I still don't totally understand, but I'm getting there.
At least you can say it. I mean, that's the first step. So, Jeff, what did you see?
Well, we saw in terms of the performance that it did meet its safety criteria by well below it was 15% MACE, and the criteria was 34 with an OPC of 25. So it was well below that in terms of the safety. When we compared it to the active controls, there were some imbalances in the groups with a very much higher rate in the treatment arm of prior strokes, which, as you know, leads to stroke begets strokes in the TAVR, which is probably the only real predictor. And also, the control group was small and pristine. It was like a Banana Republic election. It was a bunch of zeros. So, and most of the complications, and we learned this in terms of how we should design future trials, were really TAVR related. There was bleeds or vascular complications. So there was an imbalance. The stroke rates were similar, certainly in hospital. As a matter of fact, there was only 0.6% disabling stroke in hospital, but the aggregate we had was over 30 days. Again, I think a design flaw because we were working more off the TAVR VARC data, and we should really think a little differently for these trials. So there was a slight increase in 30-day stroke, not significantly, of course, with the device, but that was accrual of events from discharge out to out to 30 days.
Right.
We did learn about the device and technique because there was full coverage, what we would call all 3 vessels was maintained throughout the procedure, only about 60% of the time. And that was a matter of technique and training and the like. And frankly, that has actually progressed a lot more recently with the learnings that we've had from this trial.
So what are some of the next steps for this device?
Well, I think they changed the loader to make sure that it stays aligned.
Mm-hmm.
And it seems preliminary data, the feedback we're getting from Europe is that it's much more stable, and we are getting full coverage virtually universally. I think that's going to be key, and I think we need to collect, clinical data verifying that is, that can be maintained, because we do see on MRI analysis some very clever analysis done by the the guys up in Buffalo, where they actually did a gradient looking at lesion size in 100mm³ cuts, and found that as you might expect, the filter's main effect is cutting the larger lesions. And these are the lesions that don't go away on MRI, on the subsequent MRI. So there's very strong signals here, but we need to verify it clinically as well.
Jeff, where are we with the FDA on using signals like that for approval rather than hard endpoints? Where's the FDA coming down these days?
Yeah. Well, I mean, the FDA actually started this by looking at the MRIs. The problem is, we were looking at total volume. Well, are a lot of little ones as much as one medium-sized one? Probably not. And so we think I think this analysis, hopefully, and we're looking at, there are some preliminary data indicating that there's some good clinical correlates with the larger ones and not with the smaller ones. And we're working that up, we're fleshing that out with more data, and we'll see where that goes. And that may help shift our focus in the MRIs to something maybe more clinically relevant than just a global number.
Well, this is interesting. I mean, the regulatory science evolves as does the device, right? So it's a rapidly changing field.
Yes. And it's, intuitively, this makes perfect sense. From the early days of TAVR, we said, okay, let's just stop the debris. But we haven't had a real positive trial yet. And I'm certainly looking forward to that. Boston is, as you know, doing a 2,000 patient trial, looking at clinical endpoints. Oh, shoot, sorry.
Yeah, well, we may need bigger sample sizes to see something.
Yeah, well, the problem is these are small companies, as
right?
You know, and the FDA does try to accommodate realistic, realistic goals given, budgetary constraints. Right. And these have only been several hundred patient trials. So for really clinical endpoints, you do need, 1,000, 2,000 patient trials. But we do have one in the hopper, so we'll see where that goes.
All right. Well, Jeff, congratulations. Look forward to seeing what you find with the next iteration of the device.
Okay. Excellent. Good seeing you.
And thanks everybody for joining us here virtually from TCT 2020.